Hi everyone, this is group 5. I'm Noelle. This is Felipe, Rihanna, and Mia presenting today. We will focus on the Lassa virus pathophysiology. I'll be introducing the virus and going over its virology in the next slide. Lassa fever is a serious hemorrhagic illness brought by the Lassa virus, an RNA virus. West Africa is where it is an endemic, especially in Guinea, Liberia, and Nigeria, and Sierra Leone. Between 100,000 and 300,000 infections occur annually, resulting in thousands of faculties. In this area, the virus poses a serious threat to public health. In the next slide, we talk about the Lassa virus being a member of the mammalian virus gene and the arenavirinae family. It shares similarities with other hemorrhagic viruses that causes serious illness, such as machupo and junio. Comparing it with other related infections is made easier when we understand its categorization. In this next slide, we talk about the Mastomys natalinus rat, which is the Lassa virus main host. Humans can contract the virus by coming into contact with infected food, rodents' urine, or feces. Humans can transmit the virus through bodily fluid, and once they have contracted it, failure to follow infections control procedures puts healthcare environments at danger as well. As you can see in this diagram, it shows the primary reservoir, the Mastomys natalis, going to the rodent to human transmission, which is contact with urine, feces, or contaminated food, which causes the nosocomial, which is the hospital transmission through inadequate infection control. And then the rats also causes the human-to-human transmission via bodily fluid through blood, saliva, and urine. In the next slide, we talk about the Lassa virus and the structure of Lassa virus. The Lassa virus has two genetic segments, L and S. This is the enveloped RNA virus. The virus can enter host cells thanks to the glycoprotein in GP1 and GP2. As you can see in the image also over there, you see the enveloped virus with two RNA segments, L and S, and the glycoproteins GP1 and GP2, which helps the virus enter the host cells. The L protein is necessary for viral replications, whereas the nuclear protein shields the viral DNA and aids in immune evasion. You can see that below in the image where it shows the nuclear protein NP, which protects the RNA, and the L polymerase, which is responsible for the viral replication. In the next slide, we talk about the replication cycles of Lassa virus, which is the attachment and entry, where the virus enters through endocytosis after GP1 attaches to host receptors. You can see that also in the image. In the orange box, it points to the GP1 binding to the host receptors, which is the orange things binding to the blue host cells. And then the second step is the uncoating and transcription. After being released, the viral RNA is converted into proteins by transcription. Then the replication, which happens when the viral RNA is being replicated by polymerase L protein. And then the fourth step is the assembly and budding, the creation and discharge of new viral particles. So in all, the Lassa virus replicates in multiple steps. It enters by binding to the host receptors through GP1. The L protein within release transcribes and replicates the viral RNA. Ultimately, the fresh virus particles come together and leave the host cell to spread in neighboring cells. In the next slide, we talk about immune evasion strategies. So the Lassa virus prevents immunology activation by suppressing the type 1 interferon, which is IFN response. The Lassa virus also is the Lassa virus is capable to elude the immune system is one of the reasons it is so hazardous. Our body typically recognizes viral RNA and triggers inference reaction, but this recognition is blocked by Lassa nucleoprotein, which lets the virus multiply before the immune system reacts. So the NP protein delays the immune response by blocking the detection of viral RNA. The virus can also spread before symptoms show up because of immune suppressants. In summary, the Lassa virus is an RNA virus that is mainly transmitted from person to person and by rodents. It is especially deadly because its capacity to elude the immune system. The development of vaccines and treatments depend on an understanding of its virology. Next slide. So now we're going to start talking about the cell entry mechanism. It's GP1 attaches itself to an A dystrologican, a receptor found on a large number of human cells. You can see that in an image right there with the dystrologican being the yellow particles or image right there to your left of the image. Then the endocytosis, keratin-mediated endocytosis, is a process by which the virus is absorbed. Endosomal fusion is the GP2 initiates membrane fusion in an acidic environment, and RNA is released for replication into the cytoplasm. The Lassa virus in summary, the Lassa virus attaches itself to a receptor known as alpha dystrologican. Using its glycoprotein GP1 in order to enter host cells, the virus enters the cell through endocytosis after attaching. GP2 causes membrane fusion inside acidic endosomes, which let the virus RNA leak into the cytoplasm. This exact and successful entrance mechanism contributes to the explanation of the virus' remarkable ability to proliferate through the body.
Good afternoon. I'm Felipe Crones, and I will be covering the clinical presentation and pathogenesis of the Lassa virus. So, the Lassa virus will typically have an incubation period of 7 to 21 days. This is likely due to its immune evasion strategies, as mentioned before, with the NP protein as well as Z proteins, which we will discuss later. Once people are infected with the Lassa virus, they will typically be either asymptomatic or have mild symptoms. However, 20% of individuals will experience some form of severe symptoms. This can include, as said before, Lassa fever or a form of hemorrhagic fever or multisystem disease, which is the infection of multiple different organs and likely inflammation of these organs, which can also lead to organ failure, as well as severe vomiting and diarrhea, which can lead to dehydration and decreased effectiveness of the body's immune system to be able to fight off infection. Interestingly enough, however, despite whether or not facing severe or mild symptoms or even asymptomatic symptoms, um, 1/3 of individuals infected with Lassa virus will experience some form of hearing loss, whether it be singular ear hearing loss, like slightly less ability to hear, or complete deafness. Next slide, please. Back to cellular entry and how this impacts the infectivity of the Lassa virus. So the Lassa virus has multiple lineages. Confirmed lineages, there's 1 to 4. However, there are also assumed to be 5 to 7 other, not 5 to 7 other ones, but 3 additional ones, being the lineage 5 and 7. But there's not been much research on these lineages. What we do know about these lineages is they have very high genetic variability very high genetic variability between the glycoprotein 1, which makes it very hard for our immune system to be able to effectively attack these viruses, since depending on which form of Lassa virus lineage you got, there is now a different antibody that would have to be produced to be able to target this virus. Though they have different forms of glycoproteins, they still typically bind to the alpha dystroglycan receptor and do endocytosis into the cell mediated endocytosis. And then once they enter the cell, the highly acidic environment will begin to change the conformation of these proteins, leading to now binding to the lysosome associated membrane protein 1, otherwise known as LAMP1 protein, which then leads to fusion with the endocyte and then releasing of the viral RNA. And this is a negative sense RNA. And interesting enough, once it binds to the LAMP1 protein, it actually causes the pH to be not as heavily acidic, making it so that it still is able to open up and change its conformation to be able to fuse to the endosome, but also not acidic enough to degrade the cellular genome. And then once it enters the cell cytoplasm, it'll begin to hijack cell mechanisms. It's a negative sense RNA, so it'll begin to use its own RNA from the L protein, and then form nucleoproteins. Next slide, please. So now once it's producing its own proteins with its RNA-dependent polymerase, it's going to form multiple different proteins. Most importantly, the NP protein is very useful in evading the immune system, since we have very few things that are capable of detecting double-stranded RNA once inside of the cytoplasm. We have RIG1 as well as toll-like receptor 3. However, NP is able to help in degrading the double-stranded RNA produced by the viral genome, decreasing the ability of the immune system within our cells to be able to detect the presence of the viral genome, leading to a less substantial immune response. It does this through an exonuclease, degrading exonuclease enzyme function, degrading nucleotides at the end of the double-stranded RNA, and it degrading. As more NP is produced, this will lead to a change from transcription of the uh, RNA genome to changing to going through this essentially like transcription factor that stops it from going past, forming Z proteins, which now bind to RIG1 receptors. And and there are studies done where despite having promoters that would increase the effectiveness of RIG1 receptors, the Z protein still actually provided a inhibition to this, even though promoters were present. It is also capable of infecting dendritic cells and macrophages, which are the essential to being able to lead to the development of an adaptive immune response by infecting these cells as a result of binding to the DC sign receptors and then also fusing with the acidic changes leading to binding to the LAMP1 receptors, but not being able to fully be degraded since the pH changes are mitigated by binding to this receptor. And then they affected dendritic cells, which leads to the inability of showing polypeptides on the MHC1 and MHC2 class receptors for the development of T cells, which then, when less T cells are produced, there are less T helper cells produced, which in turn decrease the development of mature B cells. Next slide, please. Now, the morbidity of Lassa virus, though has severe symptoms, typically in the general population, will be 1%, so around 100 individuals out of 100,000. And however, when hospitalized, your ability, if facing severe symptoms and that'll mean you're hospitalized, your rate of mortality is now 15%, making it more likely for you to die. Now, however, another complication can be the location in which outbreaks of Lassa virus happens. In communities, it can be very detrimental if it's a large community with less resources, especially since it's endemic to West Africa. Having a large percentage of people who need to be treated for Lassa virus and these severe symptoms decrease the resources they're able to receive, leading to a much higher mortality rate, as well as if you receive a nosocomial infection from Lassa virus, meaning that you were likely already in the hospital for some other reason that may decrease your ability to have an immune response to the Lassa virus, increasing the mortality rate of nosocomial infections of Lassa virus. Another very big problem with the Lassa virus is pregnant women. Once again, this virus is endemic to West Africa. They don't have as many resources as we have in the Western world, sadly. But when you are pregnant and you get infected by Lassa virus, you have a much higher risk of facing more severe symptoms and also fatal symptoms. This changes depending on the trimester you're in and can be up to 30% in the first and second trimester mortality rates, and the third trimester has a 50% mortality rate, as well as 1 month from delivery, having a 50% mortality rate. Also, your likelihood of having a spontaneous abortion or a miscarriage is much, much higher when infected with Lassa virus. From anywhere to 95% of individuals that are pregnant and infected with Lassa virus can experience an abortion depending on the trimester, the first, second, or third trimester.
All right, my name is Mia Akins, and I'm going to be going over a research finding that was fairly recent. This was a 2024 study. It was published in Oxford Academic. It was titled "Presentation and Outcomes of Lassa fever in Children in Nigeria, Prospective Cohort Study." And based off of LASSCOPE, and what that stands for is Lassa fever, clinical course, and prognostic factors in an epidemic context. So yes, this was published within the last year in 2024. This study was conducted in Nigeria, which, as my peers mentioned before, is one of the in the area where this infection is most common. It was conducted from April 2018 to February 2023. And just to give a little bit of background, kind of expanding on what Felipe and Noel already talked about, Lassa fever is caused by Arena viridae Lassa virus, which is endemic in Western Africa. Nigeria is the country with the most reported cases, at least in recent years. The virus, as we discussed, can be transmitted human to human, but a lot of infections also come from rodents. So again, these are areas with not as many hygiene standards where there is more likely infection to pass from animal to human. The WHO has reported 300,000 cases of Lassa virus approximately each year, with about 5,000 deaths each year. Um, so that's a little bit of background, and going in specifically this study, the researchers who are from, oh, I believe they, oh, I wrote it down. I'll come back to that. The researchers who conducted the study wanted to specifically explore how this virus affects children to better inform pediatrics in these affected areas. Most studies in the past focused on both adults and children, or just simply on adults. So this is one that focuses just on children. It followed the cases of 124 children that had been hospitalized with Lassa fever. Their diagnosis were confirmed by doing reverse transcriptase and a polymerase chain reaction testing, so RT-PCR. Of the children, that the age range that it was 0 to 15 years old. 19 of the children were 12 months old and younger, and then the remaining 105 were between 1 year and 15 years. Um, in terms of treatment given during the study, all of the patients received ribavirin intravenously, which that is an antiviral medication. It's most known for being used to treat hepatitis C, but it's also commonly used to treat Lassa virus infections. And then the results and conclusions for the study, it found that 80% of the patients had a fever, 57% experienced digestive symptoms, 46 experienced vomiting, 4% abdominal pain, and there was also some less common symptoms such as diarrhea, hemorrhage, and the worst being kidney disease. So some only experienced mild symptoms that only lasted for maybe a couple of weeks, but then some experienced debilitating results such as kidney disease. Of the 124 children in the study, seven of them died, with four of them, the majority being from under the age of 1 years old. So it does seem that it is worse for the younger children. However, I guess the positive bit from the study, it found that the fatality rate was lower than in previous Lassa virus studies, which suggests that there has been some improvement in the treatment. Um, and then just one more thing, one of the shortcomings in this data, it did find that a lot of the people in this study, about 48% were also found positive for malaria, meaning they had had the infection recently or currently had the infection. So it's difficult to 100% say that all of these symptoms were solely because of the Lassa virus. It could have also been from malaria, which is also endemic for this region. Um, and then a little bit on the diagnosis. It's very difficult to diagnose because it does have a large variety of signs and symptoms. Some patients are asymptomatic, some have mild, and then some have extreme, severe symptoms such as kidney disease and also hearing loss, as Felipe mentioned. But the early symptoms of the virus are similar to influenza, so it's hard to identify. For this reason, it's recommended for medical staff to test for a panel of different viruses all at the same time to kind of rule out what it could also be. So things like malaria, which I mentioned are common, that's also common in that region, as well as influenza, just testing for all of those viruses at the same time, whenever the patient comes in to figure out exactly what it is. Um, Different diagnosis methods could be antigen, as mentioned, or genetic testing. Later in the infection, antibodies can also be tested for, but those can sometimes don't appear until the second week or even later. So it can be used later in the infection cycle. And as we talked about before, the RT-PCR testing has also been successfully done to identify Lassa virus infections. And then moving on to the treatment, unfortunately, there's not extensive treatments available for the Lassa virus. Um, as discussed before, ribavirin is an antiviral commonly used to treat Lassa virus infections. Other treatments that have also been explored include another antiviral, vavipiravir, which has also been used, and two images are shown on the screen of what those might look like. Um, Ribavirin and favipiravir have been used in combination for a handful of patients who have all survived their infections. However, there's not been extensive research to see what the effects might be of using these drugs in combination, whether it's better results or if it could be potentially, dangerous other effects. Other future treatments that have been explored but have not yet been solidified as viable treatment options would be monoclonal antibody combinations, as well as convalescent plasma. That's where you're using the blood or the plasma from a patient who has survived a Lassa virus infection and then giving it to someone who is currently has the infection. Again, those two things are still being studied to see their effectiveness and safety. Overall, there are fairly limited treatment options and more research needs to be done. So that kind of segues us into our next segment about vaccines.
Hi, I'm Rihanna, and I'm going to be covering the final section of our presentation, which includes the vaccine research, prevention strategies, and our overall conclusions about Lassa virus. So one of the biggest issues with the Lassa fever is that despite its high health burden and frequent outbreaks in West Africa, there is currently no licensed vaccine available to prevent this infection. Not having a developed vaccine puts millions of people at risk every year, especially those in countries like Nigeria, Sierra Leone, and Liberia, where the virus is most prominent. Fortunately, vaccine development is ongoing and there may still be some promising candidates. One of the most advanced candidates is INO 4500. This is a DNA-based vaccine that was developed by Inovio Pharmaceuticals. This vaccine, as of now, has successfully completed its phase 1 clinical trials and showed promising results in terms of its safety and immune response when tested. Another approach being explored is using recombinant VSV-based vaccines. Here they use a viral vector similar to the one used in the Ebola virus vaccines. Since this path has already been proven successful in protecting against another similar hemorrhagic fever like Ebola, there is optimism that it could be applied and work for Lassa as well. Additionally, the Coalition for Epidemic Preparedness Innovations, or CEPI, is providing major funding and support for Lassa vaccine research, which is very necessary. They have partnered with institutions worldwide to test and accelerate potential vaccine candidates. However, it's still important to understand that with the support being provided, vaccine development still takes time. It often takes many years and faces several unique challenges. One major hurdle that shows up when producing vaccines is the high genetic variability of the Lassa virus. There are several different strains. They circulate different parts of West Africa, which means that a vaccine needs to be broad protection and applied in different areas, not only just with one variant. Another challenge is the logistics of conducting the clinical trials, especially in regions where the outbreaks are unpredictable. The healthcare infrastructure tends to be limited in those areas. It's not always possible to enroll large numbers of participants to ensure consistent follow-up in rural areas. Lastly, the Lassa fever is classified as a neglected tropical disease, which often means that it has not received the same level of attention or funding as diseases that affect maybe higher income countries. This is what limits the funding and slows down the entire research pipeline, from the lab studies to the human trials to then developing the vaccine. So while there is definite progress being made in the vaccine development, none are ready for widespread use yet. This makes prevention efforts even more critical. Next slide, please.
Yeah, and kind of on that note, I wanted to briefly mention something that is very relevant and recent in the news. So this was announced on March 17th, so less than a month ago, that there will be NIH trials for a new Lassa virus vaccines. This is actually happening in the United States. So the name of the vaccine is Lassa RAB. So it is based off of a vaccine that it was developed at a team at Thomas Jefferson University. The participants are from University of Maryland School of Medicine, which is in Baltimore, Maryland. And yeah, this was again announced very recently, less than a month ago. The trial will be using 55 healthy adults. There will be 2 injections given 28 days apart. And kind of the background on how this vaccine came to be, the Thomas Jefferson University researchers published a study back in 2024 that found that non-human primates that were injected with the Lassa RAB vaccine were protected when they were later exposed to the virus. So it has a very high efficacy. So this has given the justification for a human trial to move forward. This is, an exciting treatment. As discussed before, there are, hundreds of thousands of Lassa virus infections, and currently there's no limited treatment for it. So hopefully with this new vaccine trial, once it's able to make it to the market, this will help save thousands of lives.
Okay, so because we don't yet have an approved vaccine, prevention becomes the most effective strategy that we have to fight against Lassa virus until we do get that vaccine. So I've outlined the main prevention strategies as sort of a chain of protection, which works across first like household, healthcare, and then public health. So at the household level, prevention starts with limiting exposure to rodents. These are the host of the virus and are commonly found in and around homes, especially in rural areas. People are encouraged to store food in sealed rodent-proof containers. They are also encouraged to keep living areas clean and properly disposing of trash to reduce these rodent infestations. It's also recommended to avoid consuming rodents as food, which can be a cultural practice in some communities, but practicing good hygiene, such as hand washing and sanitation, helps reduce contact with contaminated surfaces. Moving on to the healthcare level, this is where prevention becomes especially critical in the case of controlling human-to-human transmission. Lassa virus can be spread through direct contact with bodily fluids of infected individuals, and this is critical during hospital care. To prevent this, healthcare workers must use personal protective equipment or PPE, which includes gloves, gowns, masks, and so on. Hospitals should also follow strict infection control protocols and isolate patients as soon as possible to prevent the virus from spreading within the facility. Finally, on the public health level, there's a need for coordinated community and government responses. And this includes launching health education campaigns to raise awareness about how the virus spreads and how to protect oneself. It also involves training local healthcare workers, ensuring the availability of PPE, and setting up surveillance systems to detect and respond to new cases quickly. Contact tracing and early isolation can significantly reduce the spread of the virus in outbreak situations. It's important to remember that prevention isn't just the responsibility of one group, it's a shared effort and relies on individuals, healthcare professionals, and public health systems working all together. Next slide, please. To close out our presentation, I'd like to highlight the 3 key takeaways about Lassa virus. So first, in terms of virology and transmission, this virus is a single-stranded RNA virus in the arenavirus family. It's primarily transmitted to humans through exposure to infected rodents, infected rodent urine or droppings, which is often through contaminated foods, surfaces, or air particles. In more severe outbreaks, like we saw, human-to-human transmission can occur, especially in hospitals or households where there is close contact with these sick individuals. This makes the virus particularly dangerous in healthcare settings, especially those that lack adequate infection control. Second, the Lassa virus causes a serious and often deadly hemorrhagic fever. While some people infected with the virus show mild or no symptoms, in other cases, others can experience severe illness, including bleeding, respiratory distress, and even organ failure. The mortality rate is especially high for hospitalized patients. Treatments are limited, and the antiviral drug ribavirin, like we saw, can be helpful, but only if it's administered early in the disease. There's also no standardized rapid diagnostic test in many areas, which makes the treatment more difficult since they won't be able to diagnose the virus. Third, most importantly, until vaccine is approved and widely available for our defense against Lassa fever, our best defense would be prevention. This includes rodent control at the household level, protective protocols in healthcare settings, and public education and preparedness at the community and government level. With great investment in research, infrastructure, and international support, we can strengthen our response to Lassa and reduce its impact on vulnerable populations. Next slide. Thank you for listening. I hope this presentation has helped better understand not just the science behind Lassa virus, but also the public health threat that it poses. We were group 5, and thank you.